Hi everyone, this video is an introduction to proteins and their role in life science. Recall that the monomer for proteins is the amino acid, and looking at this generic amino acid, you can see the following features. There's an amino group, a central carbon, and a carboxyl group. Off of that central carbon, you'll see a hydrogen, and perhaps the most interesting part of the amino acid, this R group or side chain. So for each of the 20 amino acids, this R group here will be variable. Now, if you look at this figure that shows the biochemistry of each of the amino acids, you can note that there are several different categories of how the amino acids behave biochemically. So you're not responsible for memorizing the structure of each R group for each amino acid, but I would like you to be able to use this figure for the rest of the year, basically. So when you look at this category over here, this group of amino acids is nonpolar. There's another group of nonpolar amino acids here. The difference between these two, which is not important for us, is that this aliphatic word right here means that they're basically linear. The atoms that form the R groups are linear, whereas over here with these aromatic rings, these ring structures mean aromatic, but that's not important for us. I want you to know that these behave in a non-polar way. That is, they're hydrophobic and do not interact well with water, whereas this group over here is polar and interacts well with water. And then you have the charged amino acids over here that can behave in a alkaline or basic way. Okay, so you'll see things like aspartic acid, glutamic acid right here, and then you can have lysine, arginine, histidine. But again, we're not at the stage where you're going to be memorizing these structures. Just be able to understand that the nonpolar amino acids behave in a nonpolar way, the polar in a polar way, and you would have a figure like this to work with in the future. So when you're constructing a protein, which again is a polymer, it's a long chain of amino acid monomers, it's going to be the same reaction that has brought together monosaccharides when making a polysaccharide and nucleic acids when bringing together nucleotides. So here you can see this amino acid here is going to be brought together with this amino acid here through the dehydration reaction. So this carboxyl group here of this amino acid will interact with this amino group over here of this amino acid, and it'll result in the production of a water from this hydroxyl and this hydrogen coming together. And here you see a peptide bond has formed. So now you have a little dipeptide on the way to becoming a protein. Now in class we discussed the structure of proteins. We discussed 4 levels of structure. This is bovine serum insulin. So you can note here it has 2 chains, which is not necessarily so important for us, but the primary structure of every protein is the sequence of amino acids in that protein. So for example, here for the A chain of this insulin, you have a glycine and so on and so forth down the line. So these are the 3-letter abbreviations for each of the amino acids. Again, you're not at the stage where you're going to have to memorize those just yet, but understand that the first level of structure of a protein is the sequence of the amino acid monomers of that protein. Now, in certain instances, we haven't covered mutations just yet, but based on your understanding of the biochemistry of amino acids, hopefully you understand now that if you were to change an amino acid, so for example, if you were to change the glutamic acid that should be here for this protein and substitute it with a valine, that you might get some errors in how that protein behaves. So this is an example of sickle cell disease, wherein just a single amino acid change can create a protein that leads to problems down the road. So we'll cover this in much greater detail when we get to our gene expression unit, but I really want you to understand that that sequence of amino acids, the primary sequence of amino acids, is really important for creating a certain structure and function of a protein. Should that structure change, should the primary sequence change, you could alter the structure and consequently the function. We see this in a lot of diseases. So normally hemoglobin does not clump together and does a good job of transporting oxygen throughout the blood. But when you have this amino acid substitution to a valine amino acid, you get sickle cell. And so if you were to look at blood samples under a microscope, just by changing that one amino acid in the primary structure of the protein, you get some red blood cells that, when exposed, when deprived of oxygen, become crescent-shaped, okay, resulting in a whole host of problems for the person that has that. Okay, so secondary structure for a protein occurs because of interactions between components of the protein backbone. That is to say, between the amino and carboxyl groups, you're going to see folding occur. So, for example, this amino acid here forms a hydrogen bond with this amino acid here, and because of that, it stabilizes a certain function or a certain shape. So the shapes that they can stabilize are called alpha helices, or an alpha helix, or beta pleated sheets. Okay, and so you can see, these dots are representing hydrogen bonds. This amino group is interacting with this carboxyl group, and likewise down the helix of this alpha helix. So you get these secondary structures, beta sheets and alpha helices, due to interactions between the backbone. There's also a third level of structure that occurs because of interactions between the R groups. So you can see here, and the interactions, there's many different interactions that can occur. You can have ionic bonding between functional groups of amino acids. You can have hydrophobic interactions between amino acids. You can have disulfide bridges. You can have hydrogen bonding. So really, this tertiary or third level of structure is the functional structure of each protein. It is, for some proteins, the final shape and final functional shape for a protein. But you need to have those interactions between the amino acids, specifically between their functional groups, their R groups, to enable this third level of structure. So far we've covered primary, secondary, and tertiary level of structure. For some proteins that are made up of multiple subunits, when those subunits come together, the protein is activated and in its final functional shape. Hemoglobin is an example of that. So you can see that depicted here with these two different color subunits coming together to make a functional protein. What I want to emphasize for this slide is that, for primary structure, it's really susceptible to mutations, that is, changing the genetic code, leading to changes in the amino acids in the sequence. But when it comes to disrupting, which can have consequences in the secondary and tertiary structure, but environmental variables can also disrupt the structure, specifically at secondary and tertiary levels of structure. So we talked about heat potentially disrupting hydrogen bonds and leading to a loss of structure in the secondary level. The same thing can happen in the tertiary level. So heat, pH, they can all play a role in disrupting the secondary, tertiary, and quaternary level of structure. In class, we discussed a lot of different functions of proteins. So proteins can be enzymes, they can be hormones, they can be transporters. We will basically, for each unit that we cover this year, it's going to have a different protein that we're discussing. So we will cover all examples of protein function, but it's pretty easy to see from this list that proteins pretty much do all the functions of the cell. And so it's really important that they retain their proper primary structure, that is the sequence of amino acids, as well as their secondary, tertiary, and quaternary structure in order to be able to do what they're supposed to do. For example, act as enzymes or act as hormones. Again, thank you to OpenStax for freeing the textbook and providing us these slides, and also to Rice University for supporting OpenStax.